Good evening to all the CUET aspirants. So yes, in our previous session, what did we deal with? We have dealt with Microbial Diversity 1. Fine, right, from our CUET syllabus. In today's session, we are going to cover up, in CUET session 2, we are going to cover up Microbial Diversity 2, wherein from bacteriology, we are going to start it from sexual recombination, where we are going to learn on conjugation, transduction, transformation, followed by economic importance of bacteria, and then infections caused by bacteria, bacterial infections. Completely we are going to learn it today. Bacteriology will be finishing up today. So stay tuned for another 1 hour, one complete hour, where we will be looking into the concept in detail. So here if you see, first, sexual reproduction in bacteria, where most commonly we will be calling it as sexual recombination. Okay, this is not truly sexual reproduction because there is no gamete formation. Okay, there is no gamete formation or gamete fusion. There is no gamete fusion or gamete formation. Okay, rather sexual recombination, wherein the genetic material will be exchanged between two bacteria in any of the ways, either by transformation, conjugation or transduction. However, there is what genetic recombination, only genetic material exchange will happen. Fine, right? There is no gamete fusion or gamete formation. Fine, right? There is only genetic recombination. So most suitable term than sexual recombination is what? Than sexual reproduction, the most suitable term is sexual recombination. Fine, right? Clear till here? Any doubt anywhere, ma? Yeah. And then we'll move on to first type of sexual recombination, that is transformation. Here, genetic material is transferred from donor cell to recipient cell. Okay, genetic material is transferred from donor cell to recipient cell. Here, female there is no physical contact between donor cell and recipient cell. So here if you see, who has given out this transformation principle? Transforming principle is being given by Frederick Griffith. Frederick Griffith in the year 1928. He discovered transformation using what model organism? Model organism is Diplococcus pneumonia, okay, or Streptococcus pneumonia. Diplococcus or Streptococcus pneumonia. Very good evening, ma. Please continue with the class. If you are having any doubts anywhere, feel free to drop a message in the chat box. Any doubt regarding the subject is answered. Okay. Add-on points also you can give. So, as Diplococcus pneumonia or Streptococcus pneumonia, this is the model organism for Griffith's experiment. There are two strains of Diplococcus or Streptococcus bacteria. What are the two strains? What are the two strains? One is pathogenic strain or S strain bacteria. Okay, pathogenic strain or S strain, which is smooth strain bacteria. What is the smooth strain? Here, bacterial cell wall is covered by smooth coating. Smooth shiny coating will be there around the bacteria. Okay, smooth and shiny coating around the bacteria will give it additional support, okay, to thrive in the host environment. Okay, in host body or in host environment, the bacteria will survive because of this thick cell wall, thick shiny cell wall. Okay, and then is R strain. Second type of strain is R strain, which is non-pathogenic strain. You can also address it as non-pathogenic mutant. Here, there is no such tough cell wall around the bacteria. Okay, it just has a rough layer. Okay, the bacteria just has a rough cell wall. It doesn't have a smooth shiny wall layer like S strain. This cannot survive in host environment. In bacterial host, it will not survive. R strain cannot be survived in the bacterial host environment. S strain will be survived. So here non-pathogenic and pathogenic strain is clear right now. Non-pathogenic strain is also addressed as R strain. Okay, pathogenic strain is also addressed as S strain. Clear? In Griffith's experiment, what did the experiment say all about? Okay, what is the experiment actually? It is very important. We can expect a statement based question on the steps of transforming principle and the results of transforming principle. So listen carefully. Okay, so first what happens is the S strain bacteria, okay, first pathogenic strain bacteria is injected into the mice and mice dies as a strain can survive in the host environment. R strain, which is non-virulent strain which cannot live or survive in the host, it will die. R strain will die, and when R strain is injected into the mice, mice is alive. Okay, heat killed S strain. When S strain is heated at high degree Celsius, the smooth layer will disintegrate. The envelope, the cell wall or the envelope around the bacteria will get disintegrated, which will make the strain non-virulent. So heating up the S strain is making it what ma? Non-virulent, and when this injected into the mice, mice is alive. Fine, right? Living R strain and dead S strain. Okay, living R strain, when it is combined with dead S strain, that is heat killed S strain, the mice dies again. This says that there is something that is being transferred from S strain to R strain, which is making the R strain virulent. Okay, there is something that is being transferred from S strain to R strain, which is making R strain virulent. Fine, right? This is making the R strain virulent, and the mice dies finally. Fine, right? So what is that actually transferring is not explained by Frederick Griffith, but there is something that is being transferred from S strain to R strain, which is making R strain virulent, is what explained. Clear right now? So what is transformation actually? Without any physical contact, the genetic material is transferred from donor cell to recipient cell. This is proved by the model organism Streptococcus pneumonia or Diplococcus pneumoniae. It has two models of or two strains of the bacteria. One is S strain and R strain. S strain which is virulent and R strain which is non-virulent, and this is the methodology of the experiment, and this is the result of the experiment. At the end, what is that confirmed? That there is something that is being transferred from S strain, heat killed S strain to R strain, which is making R strain virulent. Next, here we go. Transformation of bacteria. How is that transformed? Donor DNA strand penetrates to the receptive cell membrane. Donor DNA, receptive DNA, we will call it as P plus cell. Okay. Receptive DNA strand separates and donor DNA strand will disintegrate. Can you see here? Receptive DNA strand will separate like this and donor strand is disintegrating. Pairing of donor DNA and recipient DNA, one strand will disintegrate. Nothing but he is explaining you clearly on how chiasma is being formed. Chiasmata formation happens in which stage of cell cycle ma? In Pachytene stage. Okay, in Pachytene stage. Fine, right? So chiasma like division is shown during transformation here. MC Leod, MacLeod and MC Carte in the year 1944, they confirmed that DNA is transforming material. What is that actually transferring is not said by Frederick Griffith. That is confirmed by MC Leod, MacLeod and MC Carte. The transformation occurs even when proteins or RNA were removed, but when DNA's enzyme destroyed the transformation, this confirms that DNA has no role for hereditary material. Okay, what is the experiment all about? In the test tube, they have taken the mixture of heat killed S strain and R strain. In this mixture, such 3 test tubes were taken. Okay, in one test tube, what is this mixture ma? Heat killed S strain and R strain. In 3 different test tubes, the mixture is taken. To first test tube, RNase is added, which is degenerating or degrading RNA. Fine, right? In the second test tube, protease is added, which is degrading or disintegrating proteins. Fine, right? And in third test tube, if you see, DNase is added, which will disintegrate DNA. Okay, it will disintegrate DNA. Here, when it is disintegrating DNA, there is no transformation that is occurring. It is affecting transformation in the third case. In first 2 cases, there is no effect on transformation. This confirms that DNase enzyme, as it is destroying transformation, this confirms that DNA's role as hereditary material is confirmed finally. By whom? MacLeod and MC Carte. Okay, clear till here? And you have understood this, right? What is that happening from donor chromosome to recipient chromosome in detailed steps. Clear? Any doubt anywhere from this slide ma? Yeah. And then if we move on to the next, that is competence in bacteria. Not all bacterial cells are capable of transformation. Remember that. Only some cells are competent, able to take up the DNA. Okay. Not all cells are able to take up this DNA. Fine, right? What is this transformation? If this is the bacterial culture, if the DNA is being taken up from the environment into which the bacteria is in close proximity, bacteria, it will be taking up the DNA from the environment. So here comes the concept of what? Competence. Not all bacteria can receive the DNA from its closest proximity. Only few bacteria are competent and they will be receiving the DNA. This competence is not a permanent feature, but this occurs towards the end of logarithmic growth phase. Fine, right? It is the scientist, that is Freefielder, in the year 1987, he defined competence as what? A physiological state allowing the uptake of transforming DNA related to a special protein that is present in the competent cells. Several factors can affect the transformation ability. So what is the summary that we can take out from the transformation? Competent bacteria will receive the donor DNA without any connection, and the receptive cell membrane or from cell surface, this binds to what? Receptive cell surface. DNA will enter the recipient cell, one strand will disintegrate. Fine, right ma? The donor DNA will disintegrate with the recipient's DNA. This causes genetic recombination. Genetic recombination happens in packetine of prophase 1. Clear, right? This genetic recombination is mediated by an enzyme. This is an enzyme mediated process, not energy mediated process. Remember ma? Okay, this is an enzyme mediated process, and the enzyme is recombinase. Enzyme here is recombinase. Clear till here? Yeah. So here what is the result? The rough non-virulent cells, they become smooth and virulent. Okay, rough and non-virulent cells, they are becoming smooth and virulent. Okay. So here there is a doubt. One strand disintegrate means, okay. Where did you see that, ma? Is it in the previous slide? Yeah. So here if you see, during the process of recombination, okay, during the process of recombination, here the pairing of donor DNA is happening, right? Pairing of donor DNA with recipient DNA, fine, right? At this time, one chromosome is losing its structural stability. Losing the structural stability means what? One strand of the chromosome is getting disintegrated in the recipient DNA. Now is that clear? Now is that clear, ma? Okay. The terminology says it is very simple. See, during the time of pairing, what happens at the time of pairing? Recombinase enzyme will make the recombination possible here, okay, wherein here chiasma is also formed, X-shaped structures will start beginning over here. So at this time, at the time of pairing, what happens? One strand of the chromosome will lose their structural stability, which means it is getting disintegrated. That's it. Understood now? Yeah. Cool. Okay. Now, next process we are moving on to second type of sexual recombination, that is conjugation. Okay, listen it carefully. From basics to advanced level we are dealing, ma. From basics to advanced level. Please listen it clearly. Okay, this is discovered by Liderberg and Tatum in the year 1946. And for conjugation, the model organism is what? Escherichia coli. Escherichia coli is the model organism. DNA here transfer. DNA transfer will occur either via physical contact between the bacterial cells. Okay. Here the physical contact happens. In transformation it doesn't happen. Okay. Okay. So here the transfer is happening. It is because of the physical contact between donor DNA and the recipient DNA. To understand the conjugation betterly, you have to know few terms. Here we will come across few strains. One is F plus strain and one is F minus strain. When we are looking into the cell structure of bacteria, we have come across two types of genetic materials. One genetic material is addressed as bacterial chromosome. Fine, right? One is bacterial chromosome. One is extra chromosomal DNA or plasmid DNA. Okay, one is extra chromosomal DNA or plasmid DNA. If plasmid DNA is having F factor, okay, if it is having F factor, that is addressed as F plus strain. If the plasmid DNA is not having the F factor, it is addressed as F minus strain. So this is plasmid DNA. In few of the bacteria, in the circular chromosome only, in the bacterial chromosome only, F plus strain will be present. In the bacterial chromosome itself, F plus strain is present. Such bacteria are addressed as HFR strain. Okay, in bacterial chromosome itself, if F factor is present, that is addressed as what? HFR strain. If the F factor is in plasmid DNA, it is addressed as what? F plus strain. If it is present in the bacterial chromosome, it is addressed as HFR strain. If F factor is absent in the complete bacterial genome, it is addressed as F minus strain. Okay, first you be clear with this F plus, F minus and HFR strains of bacteria. So F plus is considered as a male cell. This contains F factor. F minus is considered as a female cell. This lacks F factor. Considering this as male and female cells is just hypothetical. Okay, hypothetical statements these are. Just remember that like F plus and F minus factors itself. So here if you see, F factor exists in two forms, as plasmid and as episome. As episome, just now I told you, if F factor is integrated into the bacterial chromosome, it is addressed as episome. F plasmid will replicate autonomously when free. Okay, this will replicate autonomously when it is free. This will convert to HFR high frequency recombination cell. Okay, what is HFR? High frequency recombination cell when it is integrated into the chromosome. Fine, right? Clear with F plasmid? Yeah. Now we will clearly see how the conjugation is happening. Donor cell that is F plus strain will release out a conjugation tube. Okay, this conjugation tube is addressed as pilus or sex pili or true pili. Sex pili or true pili, or it is also addressed as conjugation tube. Conjugation tube. Fine, right? Conjugation tube or sex pilus or true pili. From donor to recipient DNA, the donor cell will be giving out the pili. Here, from donor cell to recipient cell, the conjugation tube will carry the F factor. Some amount of the F factor will be carried from here to the F minus strain, and this is how here the activity of DNA polymerase and the enzymes like relaxosome and transferosome will come into picture. DNA polymerase will be synthesizing out the new strand of DNA. Okay, relaxosome and transferosome will facilitate the movement of F factor from donor cell to recipient cell. Till here it is clear. Now, F plasmid, here you can see now new donor will be coming into picture. After the transformation of F factor, you can see the daughter cell as new donor cell. Fine, right? F plasmid, and here also F plasmid will be present. So at the products, if you see in the parental chromosomes, you can see one is of F plus strain and one is of F minus strain. And in products, if you observe, one will be F plus strain and other also at the end will be F plus strain. If for suppose the question is like this, parental chromosomes are HFR and F minus. Parental chromosomes are HFR and F minus. Then the products also will be HFR and F plus strain. Clear ma? Take down clearly. This will be clear, right? Yeah. Now, conjugation process. F minus and F plus cells attach via sex pilus. Conjugation tube is formed. Here F factor DNA moves into F minus cells and this converts F plus male cell. F plus male cell will be formed. This is also addressed as transduction, where F factor integration is reversible. Remember, this is a major important statement we can expect in statement based question. Okay, we can expect in statement based question. Important statement. Okay, F factor is integrated, right? This F factor integration is reversible in conjugation. Fine, right? Just go through these points, ma. We'll restart in a minute. So here if you see, the F factor takes part in bacterial chromosome DNA with it. This altered F factor is called as what? F prime, and resulting cells are addressed as F prime male cells only. This phenomena is majorly termed as what? Sexduction. Okay. Next process is what? Transduction. So I hope everything is clear with conjugation. Is it rolling circle mechanism? Are you talking about conjugation, ma? Yeah. Here we go. Look at here once. Yeah. Usually this is horizontal gene transfer mechanism. Okay. The gene transfer is HGT, which is horizontal gene transfer. Fine, right? Okay, this is also considered as like what mechanism of DNA transfer is involved in here. So you can also say it as rolling circle mechanism. Okay, rolling circle mechanism, and this is also horizontal gene transfer mechanism as it is in between two bacteria transferring horizontally through the conjugation tube. Okay, clear? Yeah. Next, and next is transduction. So here if you see transduction, firstly gene transfer is via bacteriophage. Remember, here gene transfer is via what? Bacteriophage. Here virus infects the bacteria. This is discovered by Zinder and Lederberg in Salmonella typhimurium in 1952. What is the model organism over here? Salmonella typhimurium. And the process over here is bacteriophage will first cause infection to the bacterial cell, and that will slowly inject the viral DNA. Viral DNA will start replicating independently using host cell machinery. Sometimes viral DNA will get incorporated into the bacterial DNA. This leads to genetic recombination. Prophase and lysogenic bacteria. Okay, so prophase and lysogenic bacteria, viral DNA integrated into the bacterial chromosome, this is called as what? Prophase. Okay, once viral DNA is integrated into the bacterial chromosome, that is addressed as what, ma? Prophase. Bacterial cells with prophase are called lysogenic bacteria. Fine, right? Occasionally viral DNA separates from the bacterial genome, and that destroys bacterial DNA, which enters into what cycle? Lytic cycle. This replicates independently, and this may also carry bacterial DNA fragments. This is all about transduction. So final key takeaway if I have to give you for transformation, conjugation and transduction. Transformation is the transfer of DNA from the other sources, not from donor cell directly to the recipient cell, but from the closest environment. If the bacteria is receiving the DNA, if there is a DNA transfer that is happening, that is addressed as transformation. Conjugation is directly between donor cell to recipient cell through conjugation tube. Third is transduction. Transduction is the transfer of DNA from viral DNA to the bacteria. Okay, this is from virus to bacteria. Fine, right? So if the DNA transfer is from virus to bacteria, that is addressed as transduction. Clear, right? Clear till here? Yeah. Now, so here you can see it clearly. First, the bacteriophage is first the virus, that is bacteriophage, is infecting the bacteria. Then it is dumping the genetic material. Viral DNA is dumped into the bacteria. Then what happens? This gets integrated with the bacterial chromosome where prophases are formed, and later that will be released out as lysogenic cycle. We have two types of transduction. One is addressed as, we have three types of transduction, sorry. One is general transduction, second type is specialized transduction, and third type is abortive transduction. We will learn about that in detail. Look at it carefully. Transducing viruses. Viruses that carry bacterial DNA during infection are called as transducing viruses. They may enter other lysogenic bacteria, and this causes genetic recombination. Types of transduction. First is General transduction, this occurs when the virus carries any random part of the bacterial DNA. If any random part of bacterial DNA is carried, when this occurs when the virus carries any random part of bacterial DNA. What is specialized transduction? This occurs when the virus carries some specific part of the DNA. Best example I can give you is lambda phasis. Here viral DNA may carry adjacent bacterial genes when excised from the host DNA. And then if you see abortive transduction, this occurs when acquired DNA does not integrate with host DNA. Remember that abortive transduction is what? Here the acquired DNA is not integrating with the host DNA. This remains separate in the cytoplasm. Okay, this remains separate in the cytoplasm. This cannot replicate independently and passed only to the daughter cells. This is passed only to the daughter cells during cell division. Clear till here? Yeah. And then we'll move on to economic importance of bacteria. Fine, right? General importance. Usually bacteria will have ubiquitous prokaryotes that play a vital role in human life and in ecosystem functioning as well. They are extensively used in industry, agriculture, medicine, waste treatment, biotechnology and food processing. Both we have beneficial and harmful roles of bacteria. So when it comes to economic role of bacteria or economic importance of bacteria and harmful effects of bacteria, what you have to remember? Most importantly, their benefits and the bacterial type. Examples are very much important here. You can take the screenshots also. I have given you the direct examples with their uses. Fine, right? Just take down the screenshots and revise it as many times as possible. That will be beneficial for you. Here, first if you see production of organic acids. First is acetic acid, example Acetobacter. Lactic acid, Lactobacillus is the example. Propionic acid is produced by Propionibacterium. Gluconic acid or vinegar by Gluconobacter. Okay, organic acids are produced via fermentation pathways, that is alcoholic fermentation or lactic acid fermentation, any of the fermentation pathways. Alcoholic fermentation or lactic acid fermentation. Next, production of amino acids. Here lysine and glutamic acid, that is Corynebacterium glutamicum, flavor enhancer, is produced from glutamic acid. Fine, right? Here you remember one tip point, important point, that is Corynebacterium glutamicum is genetically optimized industrial strain, which is frequently asked, mostly asked question. Okay, what is the genetically optimized industrial strain? Corynebacterium glutamicum. So let me come back to the sexual recombination, wherein we have learned three pathways. One is transduction, second is transformation and conjugation. From there we can expect some mechanism point of questions, some statement based questions. What is the stepwise pattern or stepwise mechanism that is happening, and discoveries and model organisms over there are important. What is the method of gene transfer is important. But here when it comes to the industrial importance of bacteria, what is that whole and soul is important is just the examples and the use of that particular bacteria is important, which is purely memory based concept. Okay, more the times you revise, more the times you will be benefited. Now we will see enzyme production. Proteases, examples like proteases are produced from Bacillus subtilis and Bacillus diastaticus. This is used in detergents, lather processing, brewing and pharmaceuticals. Next, solvents and alcohols, acetone and butanol are produced by Clostridium butylicum, and ethanol from Zymomonas and Thermoanerobacter. Methane or biogas from Methanobacillus. Zymomonas mobilis, this uses Eithner Dowd off pathway. Fine, right? What pathway is it used? Eithner or off pathway. Fine, right? Pathways are not important over here, just a highlighted point I have told you. Here you see antibiotics produced by bacteria. What are the antibiotics produced? Most important slide. You have to remember the entire table. Entire table is important over here. Fine, right? Entire table is important. You have to remember whole examples. First you see bacitracin by Bacillus licheniformis, polymyxin B by Bacillus polymyxa, chloramphenicol by Streptomyces species and erythromycin, streptomycin, neomycin and oxytetracycline. All are by Streptococcus or Streptomyces species only, and novobiocin also. Okay, remember all the different strains of Streptomyces. Streptomyces, actinomycetes, major antibiotic producers they are. Antibiotics are what? Secondary metabolites over here. Clear? If you are having any doubts, you can ask me. I am giving you time to take the screenshot, and then here we go. Role in waste degradation and composting. Organic matter degradation. Here examples if you see Clostridium thermocoelum, Thermoactinomycetes and Bacillus. Fine, right? These are used in compost and manure preparation. Next if you see bioremediation and sewage treatment. Purification of polluted waters. What bacteria is purifying them? Enterobacter, Pseudomonas, Escherichia, Bigiottoa, Achromobacter and Flavobacter. These are the examples for purification of polluted water. Pesticide degradation. Pesticides include 2,4-D and DDT. Here Aerobacter, Flavobacter and Corynebacterium. These are the examples. Pseudomonas is a model organism in bioremediation process. Remember all the points are important over here. Next. Bacteria which are used as biofertilizers. Fine, right? We have different types of bacteria which are used as biofertilizers. Just go through the table. And now here if you see types of nitrogen fixation, we have free living nitrogen fixers like Azotobacter, Azomonas, Azococcus, Beijeringia. Symbiotic nitrogen fixers include Rhizobium, Frankia and Azorhizobium. Associative or rhizosporic. This include what? Isospirillum, Pseudomonas and Bacillus. Frankia will fix nitrogen in Casuarina, and then nitrogenase enzyme is oxygen sensitive enzyme. Like in the presence of oxygen, this will not happen. Okay, in the presence of oxygen, nitrogenase enzyme is not functional. Fine, right? In the presence of oxygen, nitrogenase enzyme is not functional. Its function is disintegrated or it will be inactive in the presence of oxygen. In the absence of oxygen, nitrogenase will fix the nitrogen. That is why heterocysts are the cells which will fix the nitrogen. These heterocysts have thick and tough walls which are impervious for gaseous exchange, so that nitrogenase present inside them will fix the nitrogen. Clear till here? Yeah. Plant growth promoting rhizobacteria. Important genera you see Pseudomonas and Bacillus, and their functions usually include phosphate solubilization, production of IAA and gibberellins, and pseudobenzophore production is possible. Next is disease resistance. Pseudomonas fluorescens, which is biocontrol agent. Biotechnology, here gene transfer and recombination, DNA technology. Here comes your point, very good, that CRISPR-Cas9 gene editing tool is also obtained from bacterial genome defense mechanism. Yes, that is absolutely true, ma. You can add on that. Yeah. Insulin and interferon production, example is Escherichia coli. Now, if you see genetically modified crops, Agrobacterium tumefaciens is widely used for TI plasmid, is widely used as what? TI plasmid vector. E. coli and Salmonella are used as what? Model organisms here. TI plasmid is natural genetic engineer. Harmful bacteria you see, whooping cough by Bordetella pertussis, bronchitis Staphylococcus, pneumonia by Diplococcus pneumonia, and tuberculosis by Microbacterium tuberculosis. Animal diseases are caused by, as such, if you can see, tuberculosis, contagious abortion by Brucella abortus, mastitis, streptococcus, anthrax, bacillus anthracis, listeriosis by Listeria monocytogenes, actinomycosis by Mycobacterium bovis. Next, plant diseases, rice blight by Xanthomonas oryza, black rot by Xanthomonas campestris, crown gall Agrobacterium tumefaciens, citrus canker by Xanthomonas axonopodis, potato scab by Streptococcus cabees, wilt, tobacco wilt disease is caused by Pseudomonas solanacearum. Food spoilage, industrial damage, paper pulp spoilage, all these bacteria, please kindly make a note of it. This is completely memory based concept. Okay, so with this, economic importance and harmful bacteria both are done. And next we have to go through the detailed study of archaea, followed by cell wall layers, actinomycetes, followed by, we have to learn about mycoplasmal strains like spiroplasma and, just a second, yeah, phytoplasma, spiroplasma and phytoplasma, including 2 major bacterial diseases. This we will deal in our next class. Along with these 2 major bacterial diseases and actinomycetes, mycoplasmal strains and archaea. Along with this, we will finish off complete virology, and then we will also have a major questionnaire practice tomorrow. So stay tuned till then. So that's all for today's session, and we will meet up back in tomorrow. Tomorrow we will be completing microbial diversity, and day after we will be starting the phycology chapter. Okay. So that's all for the session, ma. Thank you.